Dear students, in today's class we are going to continue from previous lecture and cover rest of the most relevant and popularly used microbiological techniques for understanding environmental problems and environmental issues. So let's get started. Okay, so what we are talking about since last lecture are the key microbial methods used in environmental problems such as bioremediation. So the question is, how do we find the right microbes? We talked about most probable number in the last lecture, and today let's talk about colony forming units. So remember in MPN, when I talked about MPN in the last lecture, I mentioned how we undergo serial dilution and until dilution to extinction. So here initially, if I plate the microbes from the first two tubes or the first tube or from the direct natural sample, I will get too many colonies, and it would look something like this: more than 300 CFU. CFU is colony forming units, by the way. So it's number of colonies basically. And remember, we assume that each colony comes from one bacteria. So if you have 300 colony forming units, you have 300 bacteria in the volume that you have put here. So what I can do is, these are too much to count, I can't count it. So more than 300 CFU, we don't count it. Somewhere here in the middle, we will have just the right amount between 30 and 300, like 100 CFU, 100 colonies, so we can count them. But towards the dilution, we will have too few, like 24. So if it is less than 30 CFU, we assume that it might just be background error, it might be contamination, or it's just too few to be statistically significant. So when we are doing cell counting by forming colonies on plate, by plating, then we want to make sure that number of colonies on our plate are between 30 and 300. Alrighty, the other technique is microscopy. So you can actually look at under microscope and count number of cells. This one is very commonly used, live dead staining. So the live cells and the dead cells can be told apart by their color, the green ones being the live and the red ones being the dead ones. So you can actually take an image like this and then count how many red cells you have and how many green cells you have. DAPI AP fluorescence microscopy is very commonly used. So again, here we can give we can stain our microbes with DAPI and API, and then we can count how many microbes we have. Now the other question is, okay, I know how to count them manually by MPN, I know how to count them by plating, I know how to count them by microscopy, and my dear students, I must have told you in previous lectures, we use also qPCR to count our microbes. Now what do we do with the enriched isolated cultures? Okay, I have enriched them, I know how many they are, now what do I do with it? Okay, so what to do with them? Pure cultures are not very relevant to real world. So let's say I took cow dung, like in previous lecture I was talking about taking cow dung and then enriching the microbes that are resistant to arsenic or arsenic tolerant. So finally, let's say I got 5 microbes that are arsenic tolerant, and then if I put them in conditions where there is plenty of arsenic, I cannot expect them to thrive in a real environmental challenge. And there's a very nice cartoon that my advisor used to use, and I'm very fond of it. So please take a look here. These are Pseudomonas putida strains that were grown in the lab, and when they come out in the real world to clean up the mess that they were cleaning very happily in the lab, they realize that outside it's really difficult. So it says, oh dear, I did not realize in the field would be like this. We should have stayed in the lab. So we have to understand that pure cultures are not very relevant to real world. Unfortunately, in many parts of the world, people still buy and sell pure cultures believing that they will do the job, but only realize that in lab they are much more efficient than they are in real world. So the question is, how do I characterize the complex real world microbial communities? So if you look here, these are very easy to grow on plate. They will form similar kind of colonies. I can count them, I can stain them because I understand, I can count them under microscope, I can even, because I know what kind of food they like to eat, I can do MPN based techniques. But what do I do with these fellas here who are more environmentally relevant, who are more likely to be the ones that will survive in the environment? How do I characterize them? How do I enumerate them? So one method is profiling method. It gives me an idea of the dominant microbes present. So most profiling methods, they don't have very high sensitivity, so I get an idea of the microbes that are overall present, such as cloning, triflip, DGGE, PCR, NGS. NGS is very sensitive nowadays, so I can get an idea of NGS is, by the way, next generation sequencing. I can also get an idea of the less dominant microbes, but triflip, for example, DGGE, I have already talked about it, they give me an idea of the dominant microbes. And then I have fingerprinting. Fingerprinting will give me a visual profile of microbial communities that I can use for comparing two different microbial communities. Again, triflip, DGGE, PCR, ARISA, they give me, I have put NGS here. NGS does not give visual patterns, so I should remove it, but we can create visual pattern from NGS data. Alrighty, and then we have next generation sequencing such as Illumina, ion torrent, pyrosequencing, nanopure sequencing, and we can sequence the amplicons of interest. So dear students, I have said this before, but I am going to write it again so that you drill it in your head, because this is the single most important genetic marker for enumerating how many bacteria we have. (16S) rRNA gene not only tells me the number of bacteria in a sample, but it also helps me understand who is present. So if I can get like 1000 base pair or 2000 base pair sequence of 16S rRNA gene from my sample, I can align it with the well curated databases and I can find out who is present. And if I do a qPCR on it, quantitative polymerase chain reaction. So if you go back to previous lectures, I have talked about qPCR. I can also get an idea of the number of bacteria present. So in next generation sequencing, for microbial community analysis, I can amplify 16S rRNA. So instead of doing qPCR, I can do PCR on it, and then I can sequence it. And once I have sequenced it, as I mentioned, we can align it with known databases and we will get a microbial community profile. So let's look at fingerprinting techniques. Most of them are visual. So here you have a DGG gel, I've already talked about it, and here you have single stranded conformation polymorphism. The difference between the two is here, as I mentioned in previous lectures, that we have the gradient of a denaturing agent. So DNA being negatively charged is pushed away. So here we have put our samples, and now because it's negatively charged, it's pushed away from the well and it flows in these straight lanes, but as it flows it encounters higher and higher concentration of denaturing agent, so they undergo denaturing, and once the DNA fragment undergoes denaturing, it will not run away from the negative charge anymore. So whenever they are completely denatured, they stop there. Now the denaturing process depends upon the GC bonds. So if there are more GC bonds, it will travel longer because it will take longer to break the triple strong bonds of GC. And in SSCP we get a similar information, but instead of getting bands like this, we get electrophorograms, and ideally each of these peaks talk about one particular kind of bacteria, and then I can compare these peaks between sample and get understanding of alpha and beta diversity. Now all of these fingerprinting techniques, whether it is DGGE or SSCPE or RF triflip, the first step is always extraction of DNA. So I take my microbes, my cell, my environmental cell, and I extract DNA. So extraction of DNA, the three major steps of DNA extraction are cell lysis. So you lyse the cell wall so that you can have DNA in suspension, and then we capture and clean the DNA. And this is usually a multi-step process, and then we elute the DNA, typically in either in deionized molecular biology grade water, or we do it in a buffer that will keep it DNA safe for a long time. So once I have my DNA, I can do PCR. So I amplify the zone of interest. Typically for DGGE, especially if I'm or for SSCP, if I'm interested in characterizing microbes, like who is present, then I do amplification of 16S rRNA gene. And then I run a gel. This is gel electrophoresis. I run a gel and I notice if my amplicon is the right size, because sometimes I might get more primer dimers. Okay, I just talked about primer dimer, so I think it's a good idea for me to introduce you to what primer dimer is. So when I do my PCR, sometimes what the forward and reverse primer will do, instead of attaching to my DNA, they will attach to themselves and they will make primer dimer. So primer dimer, and they will keep amplifying themselves. This typically happens when I have a low DNA starting concentration compared to the concentration of primers. So I will check the quality of my PCR product, I will check the yield, I'll check the amount of primer dimers I have here. If everything looks perfect, like how this gel looks perfect, then I will run DGGE or TGGE or whatever else makes sense to me, and then I can get a fingerprint. And whatever bands look interesting to me, I can cut them out, I can elute their DNA, and then I can sequence it, and or I can amplify it, and I will know what is present here. So this is the sequence. Okay, so the typical steps for microbial community fingerprinting in summary are extract DNA or RNA if you are targeting active microbes, because remember, DNA, even in a dead microbe or in a microbe that is in a spore form or that is not very active, you can get signal, you will get DNA signal, but RNA will be only present in active alive microbe. And then I can amplify the gene of my interest using PCR. Either it can be 16S rRNA gene if I want to know who is present, or how many different kinds of microbes are present. But if I am interested in functional gene like sulfate DSR, which is specific for sulfate reduction, or MCRA, which is specific for methanogenesis, so I can amplify these genes, and then I separate my PCR products, ideally by some proxy for DNA sequence or by DGG using a denaturing agent or SSCP, and then I can do my DNA sequence analysis. So this is my DGG gel, and you see how in each lane they have run in duplicates, and the duplicates are very similar to each other and usually distinct from their neighbors, and I can get an idea of how the samples have changed with time. Okay, we can do similarly, we can do capillary based fingerprinting, and now we have next generation DNA sequencing. So this is actually a figure from Hong Wang's paper in Environmental Science Technology, and she did pyrosequencing of 16S rRNA genes that were from pipe biofilms, and after she generated sequences, she analyzed them, and this is MDS plot, by the way, and you can see that the dots that are closer to each other, they are microbes that are similar to each other. So the microbial community, all the bacteria that were present in the cement pipe with water age 5.7 days, this similar to the cement pipe biofilm that had water age of 2.3 days, and all of them are seemingly clustering together. So, and all these are chlorinated and these are chloraminated. So we can do analyses such as MDS, we can draw hierarchical clusters like I talked about in the last lecture. You can do all these analyses from your sequencing data. So this is more of understanding the beta diversity than alpha, and then clustering the similar microbial communities together. So the three most commonly used statistical techniques while doing the microbial community analyses are So the three techniques that are used, or statistical techniques often used for microbial community analyses, are clustering, MDS or NMDS, and PCOA. So clustering clusters the similar microbial communities together, and I can also get a p-value that will tell me how significant the clustering is or is not. In the last lecture I told you about some statistical packages that are used for clustering. The same in software R also has packages for doing MDS and NMDS. Basically these are dimensional scaling of our microbial community sequences, and they again do the same thing whether you do it in 2 dimension or in 3 dimension. They give you an idea of what different kinds of microbial communities are present. The closer ones will be represented by dots that are closer to each other, the ones that are more dissimilar to each other will be represented by dots that are far away from each other. And we can do PCOA, principal component analysis, or it's also called as PCA. This is very helpful when I am trying to understand how different factors affect my microbial community. So I might have, let's say I have set up a very well designed experiment, and I have different factors that I am varying, such as temperatures, pH, pipe material. So I have 3 factors that I am varying in my experiment, and for each of these 3 variations I have microbial communities that are distinct from each other. So once I have derived the sequences for microbial communities that were grown at different temperature, pH and with different pipe materials, so these are biofilms by the way, then I can do PCOA analyses and then notice how what effect of temperature, pH and pipe material was there on the microbial communities, which was the more important one, under what conditions which factor was more important. PCOA is also used, actually it's currently being used in by some research groups in USA to understand the source of microbes. So this is very much like the source apportionment studies done in air pollution, where I know the characteristics or the signatures of different sources in the environment, and I know the signature, or let us say the composition of air pollutant in the city, and then I can do source apportionment and get an idea of how much pollutant and what kind of pollutant came from different sources. So similarly PCOA, let us say I have microbial community analyses of tap water. I know that what the tap water microbes are, and I know what kind of microbes are present in the soil, in the sewage, in the droppings of wild animals, and what kind of microbes are growing in biofilm, and what kind of microbes are being discharged from the water treatment plant. Then I can do a source apportionment study to understand what component of tap water microbiome is from different sources. Who is more responsible? Is it the litter dropping of animals that somehow infiltrated into the pipe system, or is it the biofilm growth that is the culprit? So the source apportionment studies can be done using these statistical techniques. Now dear students, keep in mind that these are not the only 3 techniques that are used, but these are the 3 most popular ones, and if you plan to do environmental microbiological studies, it's a good idea to familiarize yourself with them. Now staying with statistics here, when you generate next generation sequencing data, or like the slide I showed you had pyrosequencing data, or you do Illumina sequencing, ion torrent sequencing, or whatever kind of sequencing platform you use, nowadays we tend to generate thousands if not millions and millions of sequences per sample, and when we do that, it is not very easy or intuitive for us to make sense which is a good quality sequence, which is not. So in the previous lecture I just showed you a computer, and the computer is making sense out of it, and it's telling you what it is, and then you can align it to a database. But it does not work in this black box manner where you just put it in your computer and your computer just knows. Usually there are platforms that people use to make sense of their high throughput amplicon sequencing data. So the 2 most popularly used platforms are Chime and Mother. So it's written as QIIME, but it's pronounced as Chime. And this is Mother. Both of them are free. Chime in my experience is more computationally intensive, so it requires you to install the whole environment on your computer and do. Mother on the other hand was more computationally light. So I prefer Mother, but Chime is more user friendly and makes really pretty plots. So and then if you have simple pyrosequencing data, you can use Mother, and once you derive your if either from Chime or from Mother, once you have your good quality sequences, you have separated them, you have checked for chimera, you have aligned them and annotated them, you have done everything. For your statistical work you can use R, which is again an open source software package that I prefer. People have used S, SAS, SPS and different statistical packages, but I recommend R just because it is free and it's very easy to use. Definitely do not use Excel. So use any other software package except Excel. Alrighty. So here you have once again, this is from pyrosequencing data of 16S rRNA gene of biofilms that were growing in chlorinated drinking distribution system and chloraminated water system, and the numbers on the top of these dots represent the water age, and the color and the shape of the dot represent the pipe material. So you can do some very meaningful data. If it is chlorinated, then cement pipe tend to have a particular kind of biofilms, PVC another, and iron pipe another biofilm, whereas if you have chloraminated, then they have a very different structure. So here you have NMDS plot in 3 dimensions. So you have X, Y, Z, and again by the same researcher, and the dots represent different kinds of conditions. So we have 100% GAC filtered water, 30% GAC filtered water, green no filtration, yellow is chlorinated water, and gray is chloraminated water. And in 3 dimension you can see that actually this is a movie, it's not playing, but again from pyrosequencing information you can see how the blue ones cluster together and all of them cluster together. So we notice that whether you are doing GAC filtration, which is granular activated carbon filtration, and chlorinating or chloraminating water will make a big difference in the microbial community of your drinking water. Okay, so here I have another pipeline that I want to briefly go through. And on the right, before we go through this pipeline, I want to talk about this diagram on the right. So on the right I have put the picture which is microbial community from a seawater sample, and what I want you to notice the immense amount of data that has been generated, and these are not very fine classification, but this is a dendrogram, by the way. So what I am trying to suggest by this particular diagram is that your high throughput sequencing data will be a lot of data. It's hard to visualize it, it's hard to make sense out of it. So you really need to invest some time in bioinformatics, and thankfully for you we'll have 2 lectures, at least 2 lectures later in this course where I'll be walking you through the bioinformatics platforms. Okay, so on the left you have a panel where basically you have seawater here. So from seawater you can do many things. The first thing you can do is you can amplify nitrogen fixing. Let's say I am interested in nitrogen fixation and I also want to understand the community that is here. So I can look at it in a microscope, or I can take the microbial community, amplify the functional gene of interest, NIFH gene, which codes for nitrogen fixation, and I can quantify it using qPCR. I can screen it using different probes. I can amplify the NIFH gene or 16S rRNA gene or 18S rRNA gene depending on what I am interested in, and I can sequence them, and then I will get my profile. I can also use card fish. So these are fluorescent based techniques for microscopy. I can screen them on microarray. I can also directly sort the seawater microbial community using FACS sorter. Now FACS sorter is a very interesting instrument that allows us to separate the cells based on their qualities. So I can now, using my information from this pathway, for example, I know what is the fluorophore or the fluorescent dye that will attach to NIFH gene. So any microbe that has NIFH gene, in the seawater, even before doing any DNA extraction or anything, I can make this probe attach to that microbe or its genome, and when it goes through the FACS sorter, it will be pulled towards because there is a laser in the FACS sorter which will elicit the signal from the fluorophore, and I can remove the I can separate the microbes that have my gene of interest. So this way FACS sorter can be used for sorting cells, counting cells first of all, and then sorting them according to their fluorophore, according to their specificity to fluorophore. And then once I have the single sorted cell, I can do microarray analyses. I can I can analyze them because I have sorted them according to their functional characteristics, and I can do various kinds of analyses. Now here you have another very nice flow chart for doing molecular phylogenetic analyses of your sample. So you get from wet lab, you get your sample, you extract DNA, you do replicate PCR. This is very important. Okay, and I want to talk about what NC is here. So this is a very important slide, so please take some time to look over it. When I am doing my sampling, when I am doing my analyses, we use negative controls and positive controls. So negative control, my dear students, is something that should not give you a signal. Ideally it should be 0, its reading should be 0, it should give you a blank signal because your target is not present in it. So for example, if I am extracting DNA, a negative control would be extracting DNA from pure water. So I have my deionized molecular grade pyrogen free, DNA free, RNA free water, and if I extract DNA from it, I should not get anything. But if I get a signal in my negative control, then I know contamination happened. So negative control helps me, saves me from false positives. So during sampling I have my negative control, during DNA extraction I have a negative control, and then I am doing replicate PCR. Now why this is analytical replicate. So I have the same sample, but now I am doing PCR in 2 wells or in 2 tubes to make sure if they are very similar to each other, then I know that okay, I can trust my PCR or qPCR readings. Typically qPCR is done in triplicates, by the way, and even here I will have my negative control for PCR, and all of these negative controls will also be amplified, and then I can do my sequencing, and here they are showing my seq sequencing, which is Illumina based sequencing. And now when I have my sequencing, it will go through the bioinformatic pathway. In bioinformatic pathway, I will do OTU class. I will remove first of all the noise, all the poor quality reads. I will remove the chimeras, then I will classify the OTUs, then I will do data filtering. I will remove the contaminants. For example, let us say I am analyzing sludge microbial community. I should not get human cells in it, but if I am getting human cells, then most probably it is a contamination, and I can ask my computer to remove them. I can remove cross contamination. For example, if my negative control is showing some signal, what I can do is I can subtract that signal, I can subtract those sequences from all the samples, assuming that all the other samples were also contaminated to similar degree. I can also remove false index pairing, and I can do all kind of QC QA steps. And then after this I'll go through another pipeline, which is the statistical analysis pipeline, which is not represented here. In statistical analysis pipeline, I can calculate the sequencing depth. I can understand, I can make my collectors curves to understand if my sequencing was sufficient or not. I can do alpha diversity analysis, beta diversity analysis. I can do clustering, I can do NMDS, I can do PCA analysis, and make sense out of my data and answer the questions I was asking. So dear students, this is a summary, a 2-lecture summary of the popular and relevant microbiological tools that are used to address environmental challenges. Thank you very much.